A very good afternoon. Today we'll be studying about Introduction to Bioorganic Chemistry and Reaction Mechanism. So what is bioorganic chemistry actually? Bioorganic chemistry is nothing but combining the principles of both organic chemistry and biology together and focusing on how organic molecules interact with each other within any living organism. Now, scopes of this bioorganic chemistry includes study of all the biomolecules which are present and involved in biological processes, say for example, enzyme, nucleic acids, and other metabolic proteins. Now, what is the importance of this bioorganic chemistry is that it is essential for understanding the metabolic pathways, enzyme, and principle of action or molecular basis of any diseases caused inside the body. Now, application areas includes drug designing, biotechnology, and again, biochemical processes can be understood easily in case of several deadly diseases. Reaction mechanism, on the other hand, is step-by-step process by which any chemical reaction occurs, including a sequence of elementary steps or transition states also. Now, this reaction mechanism involves reactants, intermediates, transition states, and the products. Now, types if you talk of, it includes mechanism like nucleophilic substitution, electrophilic addition, nucleophilic addition, or radical reactions. Now, again, importance of this reaction mechanism could provide you an insight into how and why the reaction occurs. Moving further to biomolecules, as we are telling that bioorganic chemistry deals basically with biomolecules. So biomolecules can be defined as the molecules that are produced by organism and forms the structural basis of all living organisms. These biomolecules can also be termed as biogenic molecules, and usually it consists of carbon, hydrogen with nitrogen, oxygen, sulfur, and phosphorus, and that's why it is termed as biomolecules. Now, these includes macromolecules like carbohydrate, proteins, lipids, and nucleic acid, and also micromolecules or small molecules like primary and secondary metabolites. Now, as the term suggests here, macro and micro. So, macro means which is bigger and bulkier, and micro means smaller molecules, which include secondary metabolites and natural products. Now, the most common biomolecules which we'll be studying and which we know is carbohydrate, proteins, lipids, nucleic acids, and vitamins. Moving further to classification of biomolecules. Now, these biomolecules can be classified into two categories, that is inorganic and organic. Now, these biomolecules actually are large molecules of many atoms that are covalently bounded together. Now, it could be inorganic, which contains minerals, gases, and water, and organic, which can be categorized with examples of carbohydrate, proteins, lipids, amino acids, nucleic acids, enzymes, nucleotides, and vitamins. Next, we have biomolecules, as I discussed before. It can be also categorized based on the size, that is micromolecules and macromolecules. Smaller size and low molecular weight molecules can be termed in the category of micromolecules, where the molecular weight might range from 18 to 800 Daltons, and usually it is found in the acid soluble pool, whereas macromolecules are large size, higher molecular weight, above 10,000 Daltons, and usually it is found in the acid insoluble pools. This acid soluble pool and insoluble pool can be considered only when you are precipitating and isolating any biomolecules. Now, as examples given earlier, inorganic molecules mostly comes under the category of micromolecules, which includes mineral gases, water, sugar, amino acids, and nucleotides, whereas carbohydrate, lipids, proteins, and nucleic acids are part of macromolecules. Next, talking of what exactly is the functions of the molecules or biomolecules. So talking of different biomolecules here, protein is building block of amino acid and its fundamental basis of structure and function of cells. Now talking of functions of DNA, which is deoxyribonucleic acid and ribonucleic acid, they are building blocks of DNA and RNA and usually involved in hereditary information transfer of hereditary information or protein synthesis. Now, polysaccharides are used for storage form of energy to meet short-term demands, and lipids, usually building blocks of fatty acids and glycerol, and its storage form of energy to meet long-term demands. Now, carbohydrates, what are they exactly? It is most abundant organic molecule. Why it is termed as carbohydrate? Because it is composed of carbon, hydrogen, and oxygen. And the term carbohydrate means hydrates of carbon. Now, Carbohydrate may also be defined as polyhydroxyaldehyde of a ketone or compounds which produce them on hydrolysis. Now, carbohydrates can often be referred as saccharides or sugars. Next, function, as I already told here, they are used to meet the short-term demands of the body, and it is dietary source of energy, precursors of fats and amino acids. It is usually present in form of glycoproteins and also participates in the structural formation of cell membrane and the several other cellular functions, including cell growth, adhesion, and fertilization. Now, talking of classification of carbohydrates, so this can also be classified into four different categories: monosaccharide, disaccharide, oligo, and polysaccharide. Now, as the name suggests, monosaccharide means it contains only one saccharide unit or sugar unit. Disaccharide contains two sugar units or sugar moiety. Oligo might contain 2 to 10 sugar moieties, and polysaccharide contains more than 10 sugar moieties. So monosaccharide can be categorized, or example can be given as glucose, fructose, and galactose. Disaccharide has sucrose, lactose, and maltose. Oligosaccharide includes raffinose and styrose, and polysaccharide includes starch, glycogen, and cellulose. Talking of each and every different categories, we'll be talking first for monosaccharide. So as I told, it is simplest sugar, cannot be further hydrolyzed. Its general formula is CnH2ON. And again, this can be further classified based on the functional group. If it is polyhydroxyaldehyde, it is aldoses. If it is functional group is keto group, it is ketoses. Examples can be glyceraldehyde for aldoses and dihydroxyacetone or fructose for ketoses. Now, it can also be divided on the basis of carbon atoms present in them. Say, for example, if it is 3 carbon, it is trioses, 4 tetroses, pentoses, or 5. Next, we have oligosaccharide. As I mentioned, it contains 2 to 10 monosaccharide units and can be subdivided in the base of number of monosaccharide units it contains. Now, disaccharide, if it contains 2 monosaccharide units. Now, disaccharides can also be reducing or non-reducing. Reducing means it contains either free aldehyde group or free ketonic group in them. Say, for example, maltose and lactose. But non-reducing means it will not contain any free aldehyde or ketonic group in them. An example could be sucrose and trehalose. Next, talking of polysaccharides, so as I told, it liberates 10 or more monosaccharide units on hydrolysis, usually tasteless. Now, this can be categorized into two categories, that is homopolysaccharide and heteropolysaccharide. Now, homo means it will yield only single type of monosaccharide on hydrolysis, and hetero means it will yield you a mixture of different monosaccharides on hydrolysis. Example could be starch, glycogen, chitin and cellulose in case of homopolysaccharides, and proteoglycans or glycosaminoglycans in case of heteropolysaccharides. Talking of structures, so here as you can see, this is diglyceraldehyde, and next to it is dihydroxyacetone. Now, if it contains CHO group, it is aldehyde group. It will be termed as aldose. If it contains only carbonyl group, it will be termed as ketose, as for example, dihydroxyacetone. Now, here you have structure of glucose and fructose, where you can see the difference in the presence of ketonic and aldehyde group. As you can see in glucose, it is aldoses. So glyceraldehyde and glucose will have aldehyde group, whereas fructose will have carbonyl group. Next, we have the structure of maltose here, sucrose and lactose, where you can see that maltose will have two different glucose molecules on hydrolysis, and lactose on hydrolysis will give one molecule of glucose and galactose, whereas sucrose might give you one glucose and fructose molecule on hydrolysis. Next, talking of two important reactions here. One is condensation reaction, another is hydrolysis. Hydrolysis is nothing but lysis of water. Now here condensation means two molecules join together to form a separate molecule. So here, as you can see, two molecules of glucose combine to form maltose. This is termed as condensation reaction. So here one OH group from one glucose and hydrogen from another glucose combines to create a molecule of water, and together they form a disaccharide called maltose. Hydrolysis, on the other hand, is breaking of that bond where water is broken. Now here disaccharide maltose, if you do hydrolysis, it will split up into glucose. Okay, so this is all about carbohydrates today. Next, Okay, we will also talk about biochemical importance here. So glyceraldehyde, if you talk, it is intermediate in glycolysis along with dihydroxyacetone. Erythritose and ribose are part of metabolism, carbohydrate metabolism. Ribose will form structure of RNA. Deoxyribose is part of DNA, as you all know. Xylulose will be involved in function of glycoproteins. Xylulose is excreted in urine. Pentosauria, glucose, you all know it is fuel of your life. Mannose is used in the structure of polysaccharide. Fructose, again, intermediate of glycolysis. And D-pseudoheptulose, it's 7-phosphate and is intermediate of HMP shunt pathway in photosynthesis. We have another disaccharide, sucrose, as you use in your daily life, that is table sugar, supplying you calories. Lactose is again exclusive carbohydrate source. To breastfeed up and lactose deficiency, it is used. Maltose is again intermediate in digestion of starch. Now we'll be discussing this isomerism in the next class, which is found in carbohydrates. Thank you and have a nice day.